Welcome our first speaker of the afternoon, Borch Jensen, who's he's a founder of Gordian and a good friend of mine. We used to live together in a row house with Peter Sikora in Baltimore and had many fun evenings before he went to San Francisco. So I'm really excited to hear your talk, Martin. I will put up your slides, and please come on stage.
I don't have this yet.
And go ahead whenever you're ready.
Thanks. Is the mic on? Yeah, it is. Thanks a lot, Morten. I'm excited to be here. This is really a cool audience. Yeah, it's a pleasure to be able to present our technology. So, obvious disclaimer, I've started a company, I work there. Everyone else at the company also works there. And what we do at Gordian is briefly in vivo pooled screening, namely putting many therapies into a single animal to evaluate their therapeutic efficacy for aging and diseases of aging. And why do we do that? Because, in my opinion at least, there's a whole lot happening in aging. We've heard talks on a variety of topics, extracellular matrix, through autophagy, through caloric restriction, and hormones, and so forth. And many of these things only happen inside of an aged organism. It doesn't happen in an in vitro system. So if we want to study aging in its full complexity, and if we want to get the answer to sort of, is this a useful therapy, we really need to do that in some in vivo system. The problem, of course, is that, and people do that, we're not the only people who go in vivo, but doing work in vivo is slow and expensive. Typically, if you have one target you're interested in, you need to either find a drug that works or create a mutant mouse line or a different species, and then you get your big mouse colony together, you can give the drug to one big group, you can give a control to another big group. This whole process requires a lot of animals, a lot of time, and a lot of money, especially if you want to bring it all the way to sort of pharmaceutical IND drug development. And so that's the problem. We want to be in vivo, but in vivo is very low throughput. And so Gordian's platform, in a nutshell, is to generate a library of barcoded therapies that we have sort of different gene targets for up or down regulation, different therapeutic payloads packaged into individual viral particles. Then we take this mixture and we put it into whatever age-related disease system we want to study, for instance, the liver of a mouse. And we put a low dose of virus in so that if you look at the cells here, most of the cells, the gray cells, have not received any kind of therapy. And they're the natural sort of diseased environment that we want to study biology in. And then a small fraction of the cells have received one of many therapies, A, B, C, D, etc. And so you run independent cellular experiments inside of the full in vivo context. And in each experiment you ask, okay, we put this potential therapy in there, we upregulated this gene, we knocked down this gene, what happened? And then as a readout, we use full transcriptome, single cell transcriptomics as a phenotypic readout where we measure the expression of every gene in each of these cells. And we then, which we'll get to, put together the full gene expression in order to interpret the cell state and the each type of biological activity in those cells. So we can measure autophagy pathway separate from the oxidative phosphorylation and so on and so forth. And then we get a giant matrix that tells us, okay, you put these 100 different therapeutics into the animal, you measured their effect on a range of biological parameters, and then you can look for something like gene F here, hypothetical gene F, that has a health-inducing or rejuvenating effect on a broad range of features. No, I can't get there we go. And that's the hit of your screen. So how does this all work? In essence, there are two things that we need to combine to make this work. The first is using gene therapy as the delivery modality. And so with gene therapy, we can still hit basically any gene in the genome. But unlike with small molecules, when we put them in, one virus particle delivers one payload to one cell. And so that allows us to separate the experiments that we do even within the animal. Whereas if we're using small molecules, if we're putting in antibodies or biologics, they end up everywhere and we have no ability to track them. But by delivering DNA cargo that has a barcode in it, we can, it ends up one place and we can track it using the barcode. And then, of course, using single-cell readout is what allows us to separate the different cells that receive different interventions from each other and interpret what happened in each one. The other very important thing that this readout allows us to do is have controls inside of the same animal. And so some of the interventions that we put into the animal are inert, they do nothing. We create various different types of do nothing to make sure we're doing nothing. And then we can compare the effect of each therapy to doing nothing in the same animal. And that allows us to get around the biological variability that you always have in vivo and that you particularly have in these complex diseases. So I'm going to present some data from a screen we did in NASH, so non-alcoholic steatohepatitis, which is fatty liver turned inflamed and fibrotic. Just very brief overview of the screen, then I'll tell you about how we're analyzing it in a bit more detail and then show the results. The animal model that we used here is a purely diet-induced model of NASH, where we, for half a year, give these mice a high-fat, high-sugar diet so that they progressively develop a NASH phenotype. Or in some cases, they don't get NASH, they only get fatty liver, which is exactly what happens in human patients. Some people develop only fatty liver and don't progress, and some have adverse outcomes. So we have different animals that have these different phenotypes. We put gene therapy in for 1 month of treatment. And in this library, we had 25 different targets. Each target we were hitting twice. We had our controls, as I mentioned. We had a bunch of novel targets that we were interested in exploring and which I'll sadly keep secret from in this presentation. But then we had sort of the best positive control we can do for NASH, which is a disease that does not have an effective treatment. And that was to take everything that was in late-stage clinical trials, either presently or recently, and see what happened, what our screen would have predicted happened in those clinical trials, and then do a comparison to what did happen in clinical trials. And this screen is in hepatocytes, so we're measuring what the hepatocytes in the liver are feeling, both intrinsically and how they're communicating with other cells. So let's just talk a little bit about how do we actually measure whether they're healthier, because that's an essential part of this and something we work quite hard on. The way we do this is to define transcriptomic signatures out of the full transcriptome for each important feature of this disease. So through talking to people like our advisor, Scott Friedman, and other experts, we kind of have a list of these are the things that are going on in NASH that matter to the disease. And so that's stuff like inflammatory signaling, senescence is a driver, metabolism of lipids and of the cell in general is important, and there are specific signaling pathways that are important for NASH. So for each of these, we have sort of a custom gene list weighted in the right way to represent, yes, this transcriptome signature represents this type of biology. And then we can score for each of the different targets, and we'll come back to this. This isn't the juicy data slide. For each target, we can score, did it move each of these features towards a healthy state, or did it make it worse? And then we get the big matrix of what happened for each of these therapies. And of course, for each of these features, what we're doing is, just as don't pay too much attention to this specific graph, but we have our inert, the cells that have received controls somewhere on the axis of, one way of this is healthier and the other way is less healthy. It's the gene activity axis for this particular feature, which is senescence. And then we can see that each of these colored dots represents all of the cells, the thousands of cells that we captured with a given therapeutic, pooled together to get the average effect of that therapy on cells within this disease. And then in the case of senescence, I'm just showing one positive control here, which is if we overexpress P16, you get the cells, put the cells into a senescent state, and appropriately we do see that this intervention makes senescence worse. It creates more senescence. So we have various controls like that we use to make sure that we're actually measuring what we think we're measuring, and then we can interpret all these other interventions in light of that. This gives us our matrix of what's going on, but it is only transcriptomic. We want to have confidence that these different features that we generate really correspond to real physiology. And so for features or groups of features, such as like these various features that all contribute to liver steatosis, we can take something that seems to have a positive effect on this set of features, and then we can make a prediction. In this case, we have a net benefit on the steatotic features from putting FGF19 into the hepatocytes. Our prediction is that this would reduce steatosis, and we can then do a sort of normal experiment where we just put that one thing into a group of mice. We look at the livers with histology. White is fat here, so even if you're not used to looking at histology stuff, when we treat with inert controls, you have a lot of fat in the liver in these mice. When we treat with FGF19, you have a massive reduction in liver fat. And so in this way, we can validate with orthogonal assays each of the features that we're using in our screen, and thus gain further confidence that what we're measuring in our screen is actually representative of what would happen in the animal with saturating treatment. So we take all of this, put it together, and then we look at each of these interventions and make predictions around what would happen if we put this in alone at a therapeutic dose. And so in this case, for instance, we would expect that there's a positive effect on steatosis, not so much of an effect on inflammation, maybe a little bit. And then these various pathways that we're interested in, we also have predictions around. And so these are all clinical targets that either have been tried recently or are still in trials in NASH. And so what we did was run and analyze the screen results for each of these and then compare it to what actually happened in the clinical trial. And we did it in that order. So I wasn't looking at the trials before. I knew that none of the trials had worked, but I didn't know what the specific outcomes were. And so this is a little bit busy. On the left of for each intervention, you'll see the scores that you were just looking at. So for each of these features, what is the effect of this intervention? They're grouped into clusters that correspond to physiological scale assays. So in the liver, you don't measure each of these cytokines mostly in the trials. You just look at immune infiltration as a marker of overall inflammation. And then on the right side of each column here, you see this is what happened in the trial. The trial is listed down here. And where you see a green box is basically when we made a prediction that is roughly the same as what actually happened in the trial. Of course, you can't exactly translate a transcriptomic change to a certain size benefit, but like on the scale that we're using, they map onto each other. So FGF19, we thought that there would be some benefit in inflammation. That is indeed true, only in 30% of patients. We were more confident that there was going to be a benefit in steatosis. There was, only in 50% of patients. For FXR, that was our worst performer, where half the predictions we made were not correct. There's a couple of potential reasons for this. I think one is that we predicted no benefit in the fibrotic score. There was a benefit, but only in 10% of patients. So maybe this is just within the noise of an actual real benefit. Or maybe FXR is known to also have some activity in the gut, and so we are only measuring in the liver. But for every other intervention that we tested, when they had measured, in some cases they hadn't measured what happened in the clinical trial, but every time they measured it, we made basically the same prediction. And as I've noted here, We've done this one screen in the amylin mice that I mentioned. We've repeated the screen in the same mouse model, but with a different promoter, so a different effective dose of the gene therapy, a lower expression level. And we've repeated it in a different animal model called the fat model that some experts like, and we get roughly the same outcomes with exceptions that sort of make sense. Like in the fat model, you don't have a lot of lipid accumulation, and so the effects we see on steatosis is smaller, just because there is no steatosis. So honestly, I was a postdoc before I started Gordian, and it's not usually the case that things work this well. And after all, this is only in a mouse model. But at this point, my own skepticism has been allayed, that I'm convinced that we can predict, by going into the right animal model, we can actually predict the physiological outcomes with this platform, and therefore we can do really high throughput screens for in vivo efficacy. And so, to summarize what we're doing here, I would say we're cheating on the drug development test. Rather than moving towards, like, let's work on this drug, it has some promise, and then go and spend $10, $20 million on developing it, we can take the best animal model for each disease that we're interested in. So for instance, we are right now working in horses for osteoarthritis, because their joints are more similar to human joints and cartilage. And the horses that we're using are, they've been running around getting osteoarthritis by, like the same way that humans do. For NASH, we're moving into primates and so on and so forth. We can use the most relevant spontaneous disease aged animal model for each disease, then run our screens and map out the whole landscape of which interventions are effective, in which case. When we get something that's really good, all blue, we can move it forward. Occasionally, we might get half blue plus half blue and want to do a combination of those, which we can then do in a rational way. If we don't find anything, we just drop it and don't move forward. And then based on what the target is for in each of these indications, we can choose the ideal modality. Maybe it's gene therapy, maybe it's an antibody, depending on what the target is, and then go into the same, clinical preparation that every other company does, but already having done in vivo validation in the most relevant model of disease, and therefore we predict, and we have yet to prove, with a much higher clinical success rate. And the other thing we can do, and I'll eat into my question time with this, is I want to understand aging, right? I spent 10 years trying to do that in academia. I still want to do that. And I want to understand what's the relationship between these different mechanisms of aging that we've been hearing about and then the decline that happens with age. So what are all these relationships? And because our screen measures every aspect of the cell state, we get a score for senescence, we get a score for autophagy and mitophagy and so forth. Each of these screens actually tells us in a given disease state what is dysfunctional. And the targets that we test in each different disease state, we see whether it has an effect on senescent cells or specific features of aging. And so we have this modular system where we can start to map out, like, what is the treatment for the bigger picture of aging. So let me just zoom through the summary. If you want to study aging, ideally you study it in a naturally aged organism. If you want to do a lot in a single organism, you should use pooled in vivo screening. Pooled in vivo screening works, at least insofar as predicting clinical outcomes in NASH. And by creating a big map of what the best treatment is going to be, you can plan out your entire clinical development, pick the right modality, pick the easiest path to success, and hopefully get us aging drugs sooner. That is the end of the Gordian talk. In case anyone missed this Wednesday, I'm just briefly going to mention the Longevity Impetus Grants, which is a new initiative that's launching on Monday. And it's going to give away a lot of money to basic research into aging and longevity. There are no strings attached. It's not an investment. It's just giving money to scientists if they write great applications for work that really would make a difference and that isn't already going to be done. So go to impetusgrants.com and you can put your email and you'll get a notice when it launches on Monday, or hopefully our distribution campaign will successfully reach everyone in this room anyway. So if you're doing cool stuff, please apply. Thank you, and I guess questions, 1, 2?
Thank you. Thank you so much, Martin. Do we have any questions from the audience? We have a question here from Mike.
Yeah, nice talk. It's a very exciting technology. I noticed that you're using about 25 guides in the, or 25 overexpression targets in the example you gave. Kind of what informs that decision on number of targets, and is there kind of a calculation that you guys think you can, increase that number, and maybe talk a little bit if you have a rational, guide to pick those 25?
Yeah. We are increasing those numbers. We're assigning screens of sort of more than 100 right now. The limiting factors are number of cells in the tissue you're going into, which isn't really a big issue for the things we're doing right now, but is a theoretical limit. And then how much money are you willing to pay? Sequencing is not cheap. This is more expensive than taking pictures of cells in culture. How do you design them? In theory, you could just do whole genome from the start. I think you can be smarter about it. And so we have multiple sources of input on like what are we putting in there. A very important one is human-linked targets. So anything where there's human data from GWAS or otherwise, therefore giving us even more confidence in the clinical path. We would just test all of it. Known pathways, if you think autophagy is interesting, let's test all of it. And then also our data science, bioinformatics, sort of that we use to analyze the data. Also, it can be used to make predictions around like what is the highest leverage thing to go after. So we do that as well.
Really nice. We have another question from Vadim.
Yeah, very nice talk. Have you thought of quantifying the age of cells after following your therapy at a single cell level?
Yeah, for sure. So we're a company. I don't know what an aging drug is yet, so we're going after these specific age-related diseases. But the reason I'm excited to build this company is that the entire platform works for treating aging. You just substitute sick, healthy for old and young, and we're already using old animals, and we're already collecting the data for that. And we can, with the data that we've already collected, we can just go in and define the features of aging and then look at those. So we're already collecting that data, even though it's not sort of the commercial path.
Very nice. Do we have more questions? We have one on Slack here. There's one that's upvoted from Eva van Galen. How does this platform deal with systemic diseases when choosing a disease model might be harder? Or where choosing a disease model might be harder?
Yeah, that seems true. We're focusing on sort of one organ at a time. You could do systemic. It would just cost more money. So maybe we will do that kind of thing later.
All right. Thank you very much, Martin. Really exciting stuff.